Click for IB Biology. Here we're looking at the Crossbridge cycle, which is the mechanism of increasing the overlap between actin and myosin. So in essence, this is the mechanism of the sliding filament hypothesis, which has the shortening of the muscle is created by an increased overlap of actin and myosin. So we're looking at how that actually occurs. There's a PDF of this presentation in the description, along with some quick quiz questions with their answers and links in the two other parts of the channel. Let's take a look at the diagram. This diagram represents the Crossbridge cycle of actin-myosin interaction and the role of other molecules in that process. We will zoom in and look at each of the steps and attempt to explain how it can be that actin and myosin can move relative to each other, increasing the degree of overlap and therefore the shortening of the muscle. We begin our analysis by looking here with the myosin molecule in this location, and note the position of the bridge. This structure here is called the bridge, pointing in this angle here. This is of significance, and ADP is attached at this point. If we look up here to the thin filament, which of course is actin, we have more detail, and we can see that the light purple represents the actin, but there is also two other molecules, both proteins, one called Tropomyosin and the other called Troponin. Notice that the position at this phase in which the muscle is relaxed, the Tropomyosin is sitting over the actin molecules, and those purple, darker purple locations there are what we call binding sites. And this is where it's possible for myosin to molecularly join to actin. But this is not possible during the relaxed phase because they are occupied by tropomyosin molecules. If we move to the next phase in the Crossbridge cycle, what we see is the arrival of calcium ions, and you'll recall that these come from the T tubules, and you'll also recall that they are released from the T tubules in response to a nerve impulse. We did a video on innervation and neuromuscular junctions, which you could review to understand this further. These calcium ions bind to troponin, so that's this one here, and in turn this causes a change in the structure of tropomyosin, which is the red line here, and it moves it away from the binding sites. Here's one here, other ones there, and there. And now it's possible for myosin to bind to the actin in a molecular manner. So it's happening at this location here. So notice that the angle we have on the myosin molecule is like so. Moving on in the sequence. In the next phase, what we see is that the angle of the myosin molecule has changed. In effect, it has moved from this position to this position. This is not a joint in the molecule, this is a molecular shape. And the new shape, this new angle, leaves the myosin at a lower energy level, which is what's driving the positional change. And so what has now happened is, in this individual diagram, is the arm has moved from here to this position here, and the degree of overlap with actin has increased. If repeated along the entire length of the sarcomere and the multiple sarcomeres and the muscle, the entire muscle length has just shortened in what is called the power stroke. In the next phase of the cycle, just position this a little bit better. In the next phase of the cycle, ATP attaches to the myosin. So we might call this the contracted condition. Now ATP is going to join onto the myosin, and the combination of ATP and myosin reduces the affinity of the myosin structure for actin. So let me just annotate. So reduced affinity of myosin for actin. And of course, this is going to lead to a detachment of the myosin bridge from the actin. So let's take a look at that. I'll just reposition. So this ATP attaching to this myosin has reduced the affinity here, and so it detaches. The ATP has not been broken down or hydrolyzed at this point. When that happens, if I reposition to here, then this brings us full cycle, and the hydrolysis of ATP is to bring about the repositioning of the myosin in its original position there. So ATP is used to detach the actin and the myosin, and then secondly to reposition the myosin bridge ready for the next contractile movement. This is called the Crossbridge cycle. And there we can see the entire diagram. When viewed under electron microscopy, rather than at the molecular level, what we observe is that the Z bands here in the relaxed muscle, they become, that Z to Z distance decreases, and we now know that this is due to cross-bridge cycling between the actin and the myosin, with an increased degree of overlap between the actin and myosin here. Interestingly, if one thinks three-dimensionally of the actual myofibril structure rather than in the flat band structures here, the process of contraction is not only a shortening of A to B as shown in this diagram here, but involves a twisting action, a torsional twisting action, and with that torsion becomes the stretching of the tintin, and that's where the elastic energy is stored. Links are here to the channel and to the entire playlist, where you can find the other videos which are supporting IB biology.